Learning is a treasure that will follow its owner everywhere. I am very happy to start this video with this Chinese proverb emphasizes the importance of learning. I hope you should learn something from this video. Hi everyone, this is Mr. Johnvasanth James, Assistant Professor from the Department of Biotechnology, J College of Arts and Science, Pudukkottai. In this video, I am going to discuss about teratogenicity, a very important concept of pharmaceutical biotechnology in a very comprehensible way. Let's get started. Toxicity studies are very important in order to determine whether newly formulated drug or newly discovered drug exhibits any short-term or long-term toxicity. Teratogenicity is one of the important toxicity studies which is used to observe any significant effects of any drug in anatomy and physiology of fetus secondary to prenatal treatment. In short, it is defined as the observation of any significant change in the function and the form of a fetus. It is also defined as the ability to cause defects in a developing fetus. Now I hope you clearly understand the definition of teratogenicity. It is also important to know the meaning of teratogen, which is entirely different from teratogenicity. Teratogen is any substance or agent that can induce the fetal developmental abnormalities. Example, alcohol, radiation, etc. Now we move on to the next heading, steps involved in the teratogenicity. There are three main steps which is carried out in the teratogenicity studies. First one is selection of animals for animal study, administration of drug into the animal model. The final step is observation of animal for teratogenicity. Let's discuss all the three steps one by one. Different species have been used as animal model for teratogenicity screening, most of them sharing similar developmental process with human. Choice of animal depends on the teratogenic events being addressed. We commonly use rodents, rabbits and in vitro cell lines when our study concentrate on drug safety and teratogenicity. Monkeys, which belong to non-human primates, have also been used in the teratogenicity studies. Nowadays, research work is going on for the biological development of animals. Most used in teratogenicity test is addressed with the aim of maximize the human translation and reducing the number of animals used, and finally fast marketing of new drugs. The second step is the administration of drug into animal model. It may vary from species to species because that causes the discomfort to small extent. For example, intravenous or intraperitoneal mode of injection of drugs are opted when we use pregnant mice as the animal model. Sometimes gouging method is followed when the drug is administered directly into the animal stomach through a tube. Sometimes agent could be given to animals through its food, but it is not suitable for teratogenicity study as animal may not consume all the food in one sitting and result in an inaccurate receipt of drugs. And these are some of the pictorial representation of intraperitoneal injection and gouging methods. In case of embryonic exposure in chickens, the chemical agents can be added in a solution over an embryo. In case of teratogenicity studies in marine species, the agents can be mixed directly into water in which the embryos are bathed and analysis of resulting embryos can be carried out in different time periods. The third and final step is observation of animals for teratogenicity. After administration of drug to pregnant animal models, they are sacrificed close to term and a full autopsy on mother and fetus are analyzed for the teratogenic events. If the testing drug is found to be harmless, which refers that the testing drugs has potential to develop new therapeutic drugs and it can be safe for use by pregnant women. If the testing drug is harmful to mother or fetus, then the experiment will be terminated. Next, let's discuss some of the teratogenic medication. Low dosage of anti-epileptic drugs can cause the cognitive disorder in developing fetus, whereas high dosage lead to the structural deformities. High dosage of antimicrobial drugs can cause severe as well as very harmful effects in the fetus. Anticoagulant drugs can cause skeletal abnormalities such as stippled calcification of epiphysis and the nasal hyperplasia in developing fetus. Anticoagulants like coumarin can induce the CNS deformities. Next, the antithyroid medication can cause the aplasia cutis, that is congenital absence of skin, and choanal atresia, that is blockage of back of nasal passage, and esophageal atresia, which means underdevelopment of esophagus in fetus. In case of vitamin A, both overdosage and low dosage can cause the embryonic deformities. It can also induce the thymic and cardiovascular abnormalities such as hyperplastic aorta and cardiovascular transposition. The women who exposed in utero to hormonal medication like diethylbesterol can develop clear cell adenocarcinoma of vagina and cervix and structural anomalies in the genital tract. Not only a pregnant woman, but also a boy babies of women who received this hormone during pregnancy develop abnormalities of genital tract. Next, we discuss the FDA categories of drug used in pregnancy. The category A drug denotes the no risk in the controlled human studies. Adequate and well-controlled human studies have failed to demonstrate a risk to the fetus in the first trimester of pregnancy, and there is no evidence of risk in later trimester too. In case of category B, animal reproduction studies have failed to demonstrate a risk to the fetus, and there are no adequate and well-controlled studies in pregnant women. Or animal studies have shown adverse effects, but adequate and well-controlled studies in pregnant women have failed to demonstrate a risk to the fetus in any trimester. In case of category C, animal reproduction studies have shown an adverse effect on fetus, and there are no adequate and well-controlled studies in humans. But potential benefits may warrant use of the drug in pregnant women despite potential risk. In case of category D, there is a positive evidence of human fetal risk based on adverse reaction data from the investigational or marketing experience or studies in human, but potential benefits may warrant the use of drug in pregnant women despite potential risk. In case of category X, studies in animals or humans have demonstrated fetal abnormalities, and there is positive evidence of human fetal risk based on adverse reaction data from investigational or marketing experience, and the risk involved in use of drug in pregnant women clearly outweigh potential benefits. Thank you so much for watching this video, and I hope this video is very useful to you. I conclude this video with a quote: "The capacity to learn is a gift, and the ability to learn is a skill, and the willingness to learn is a choice." Thank you for your willingness to watch this video and learn some concepts from this. Thank you so much.